Happy to introduce my colleague from across the hall, but you know how these things work. I wind up seeing him more here than in our building, which you'll see a picture of in just a moment. Jose has been at Rice and heading this Center for Theoretical Biological Physics since
2002. 2002.
No.
At Rice.
I was going to get to that. So you moved to Rice in
2011.
2011. Okay. So
but the center moved to 2012.
Yeah. But before that, as you might know, it was in San Diego for many years, where he, along with colleagues, started this center, an NSF Frontier Center, the only one in biological physics, essentially. And only Jose could keep such a center renewed multiple times. It's an impressive feat. And I've been a beneficiary of that, I have to say, so that's great. But so today he's gonna tell us about nuclear physics, right?
Nuclear physics, yeah. Yes.
Physics of the genome. And there no doubt may be some continuation of some discussions going on yesterday. So,
okay. You see, only in the Center for Theoretical Biological Physics, the nuclear group meeting deal with chromatin. But first, I want to thank the invitation to be here. It's a pleasure. And the talk I'll give today will be not too mathematical. It's sort of bringing the ideas that we have. I hope that people can enjoy it. I have multiple sections, so I can stop at any time. So there's no reason to speed up or hurry up. We can go to the different topics. And when we moved to Rice, we met this very interesting character, Aris Aiden. Basically, he just had joined as assistant professor with this technique called high C. He was one of the guys that was the creators of this technique called high C. So basically, we thought, why don't we get to understand chromosomes? Basically, we did protein folding. Now it's just a bigger polymer. Let's go into that problem. I will tell a little bit about the stories that go behind it. So first, is basically that's where we are located on the 10th floor of this building. That is the center for that is the BRC, the Bioscience Research Collaborative. Just to have an idea, if you leave from this building, it's close to the medical center, it's about a 10 minutes walk to Baylor. It's about a 15 minutes walk to MD Anderson. It's about 25 minutes walk to the physics department. So that's Houston, and that's how we are located. And lots of interactions across the street. And I remember when I moved to Houston, Al Gilman had this program, what they call ChIP-Seq, the Cancer Prevention Research Institute of Texas, and Al Gilman was there. And after the package, they say, why don't you try to get a grant to work on cancer? And I remember we submitted a proposal, and Al Gilman gave him a call, and he said, Jose, we know each other for a long time, and you know nothing about cancer. He said, you're absolutely correct. But he said, but the people across the street know nothing about modeling, theory, data analysis. So you have to promise you're going to cross the street. So that's how I got funded on something what I accept. I don't know. Actually, I learned a lot about cancer during these years. Actually, my colleague Herbert Levine moved almost 100% into that field now. I'm still doing a lot of work on that. So to continue, basically, we heard a beautiful talk by Vivek yesterday about these things. I'm going to try to stay a little bit to the basic ideas associated to that. Chromosome is a fantastic thing. If you think about it, it's a polymer. If you get the entire chromosome is about 2 meters long, okay? You have to collapse it into a nucleus that's a few micrometers. And really, you want to avoid knots. So it basically, you figure out that's a major task. Why you don't want knots? The main reason is basically if you want different regions to be expressed and be open, you don't want to sort of have to unfold the entire thing to do it. So it's an amazing task. And if I continue here, this field has been for a long time. I think basically it's really a 2000 story, the one based lots of the great experiments appear. I think this is one of the classic experiments, the idea that came on the chromosome territories, that if you look on the nucleus, basically most of these chromosomes tend to complete, separate from each other, separate from each other, and don't mix. And actually, in case when these chromosomes start to mix or duplicate more, that's when you have lots of diseases and associated with that. So actually, this idea of being able live separately is it is an important part. And then there has been lots of experiments that start from our experimental friends where these things form loops. And I will tell you what they call about loops, domain, TEDs, or names I'll tell you about as we go around. And base here, they call each chromosome separate, they call territories, right? Just to keep the notation that people use. They didn't create that notation, but that's how it goes. Now, this experiment amazed me from the first time, particularly if you understand the chemistry of it. So the idea is you try to get a map of context, which parts of the chromosome, when the chromosome folds in 3 dimensions, they come to form a 3D space. The experiment is amazing because what you do is you try to cross-link here with formaldehyde. There are other things that people can use, but basically if things are closed in space, you can cross-link them. Okay? And then you cut the entire chromosome and just go and chop it as much as you can with these restriction enzymes. Then you put biotin at the end, you ligate this small piece. Then you really chop one more time and you get just these places they're coming together here. And as you sequence these small pieces, you can figure out which parts that are very far in sequence, they're close in space. Now, the amazing about this experiment, if you have, I'm not an experimentalist, but I have collaborated with experiments for a long time. As you can see, there's nothing to go wrong. It's amazing that it works, right? Based the amount of chemistry steps you have here and these things here, but people actually became very, very, very good. If you look, every single step here has some chemical problem. People really became a master of it. If you go to the previous slide, Ares is the guy that do that. Olga Duchenko, that's his main research scientist. She's absolutely spectacular getting these things running. And we have had a long, very fruitful collaboration in the last few years. So the idea is that from this contact map, we hope to figure out more things that are on space. And the interesting thing that really got people very, very interested on that is the following thing. Vivek gave the talk yesterday, and he told about every DNA is the same, means the genetics is the same, so you're left with epigenetics. So what differentiates cells is going to be epigenetics. And the amazing thing is people start to got these maps that are these contact maps. The more red you are, the more chance of having 2 pieces of chromosome making contacts. And you have all these patterns that appear from these contact maps. But the nice thing that people learn is that these things are actually well connected to epigenetic markers. Basically, these are things you tend to follow. They're either are basically methylated histones, right? And you start to see all these epigenetic markers. Remember that you can follow other, what they call ChIP-Seq, other epigenetic things. Histones are interesting because, as we also heard yesterday, it was good to have an introduction. They're going to associate to form more heterochromatin that tends to be denser, more compact, less active genes on it, right, as we go along. But the fact is that you have this relationship between what people call about it. Basically, you can go and try to do some clustering to figure out how many different patterns you have here, and they correlate well with areas. You see, for example, here, particular markers, they're actually associated to this white, connect this part and this part coming together. So it's an amazing, it really help people to understand this relationship between geometrical features and energetic features. Now, just for curiosity, I think is to show that basically these markers here, these are just 2 cell lines. This is basically what I told you is the cell line GM1278. That's basically is a lymphoblastoid, it's a blood cell, but it's a very stable cancer cell. And why people use it? Because for experiments, every group believes they're looking at the same thing, right? So there's nothing special about it. So it became one of the main markers. This is a pulmonary cell, and you can see they have different epigenetic markers that go with them. Absolutely. Yes. Yes, they do. We have experiments on HeLa cells. People avoid to get close to HeLa cell these days for their people make your life very miserable, how ethical you are, what you're supposed to do. So it's basically, so people have done HeLa cell. Ares has done HeLa cell. They're actually great cells to work with, right? But people just have They do if they have to, but they avoid to go to things just for political reasons these days, not for any other reason that you want to do it. So, continuing this discussion, I think that this is just what people got it. In the beginning, you heard about the different kinds of compartments you have, the A's and B compartments that are based. The A's tend to be the more active gene part, and the B's are the heterochromatin, so L-chromatin and heterochromatin. These are the things phase separate, but actually you have subcompartments. People have noticed on these things. That's the best way to represent. There are about 6 subcompartments. Actually, before existing very rare cases, most of the stuff you can do with 5, but that's the way a lot of the data has been explained these days, at least on human cells. Okay, so we have these compartments, subcompartments, and they tend to phase separate. Now, if you guys ask me at the end, you can tell why they phase separate. You're going to see that our models here are much simpler. I showed that on a slide before. Maybe I'll show the next slide. Here is, we create Hamiltonian for that. Now, the resolution we present our initial data, every bit is 50 kilobases. So when we say these things are methylate or things are acetylate, means they have a higher density compared to the other things. So it's basically completely different than the work we do on folding. These are really things. There are beads. The size of these beads, they're typical of the size about 0.17 micrometers. So it's a different scale, just to remember you're not doing on base pairs. Just if people want, for curiosity, is interested to say that this is 50 kilobases, 10 kilobases yesterday, anything on the kilobase scale or more kilobase imaging is very good. Air is coming down on it. I have a lot of trouble dealing with the data much smaller, because then you try to figure out, are you actually seeing context or you're dealing with the chemistry of it? You can look at data, but let's not get in there. Then you have lots of work that people do at the level of nucleosomes. And nucleosomes are about 250 bases. Just remember, that's the size between these things they have. If you go to yeast, they're 120, they're much closer. But then genes actually live between 500 bases to about 5 kilobase. So that's a very funny thing, because there's very little imaging actually doing on this sort of a area that you have. But that's where the field stands. There's a lot of new techniques coming out. The imaging techniques are getting better by the day. Well, that's one of the reasons I love this field, because it looks like protein folding on the 90s. Right? Basic. No, if you look protein folding 90s, just to give a story, people like Alan Firsch here at Cambridge got the idea of doing multiple mutations. Alan in scan. Now everybody does that. But when I was in graduate school 5 years before, people would do activities on a single mutation. So the fact is, you could do multiple mutations, you could do like statistical things. So now these imaging techniques are changing the field. So our understanding is going to change, because the experiments are just getting better and better. And that's a great time for theoreticians to jump in. Okay, so we're going to create a model. And our model, basically, I'm going to describe a little bit for you. It has two parts for it. I'll first talk about the second part, that's based trying to create a polymer model, where we come here. Our sequence, we are going to describe the sequence based on these A and B compartments. Actually, have the subcompartments also. So I'm going to have a heteropolymer here with these beads of these different colors. But I'm going to show that actually, if you look, what people call the ChIP-Seq information, but epigenetic information from the cell, we can learn these types. So the beauty is, we first work this part for right now. We can do everything, start just from ChIP-Seq, and we can use Hi-C just to verify if our model is actually giving appropriate answers. You don't have to use the beginning, you have to use Hi-C at least to get the first eigenvector to figure out where the A's and B's were. Now we can do all the way. So this is really like a polymer physics kind of approach to the problem. This is really a machine learning, not the most sophisticated machine learning, but it's enough, because as long as you have enough information, you just have to classify all these loci in terms of these terms. So I'll tell you. Let's tell, and I'll be glad to go in every detail here. So in the polymer model, we create a polymer that's basically is a homopolymer term, with a term that's responsible for phase separation, where these things are coming together. We have the loops. Actually, if you're happy to 50 kilobases, they're not that important. They actually give structures inside this physical base. You can run the model with or without it, depending you want. And then we have this term, we have the ideal chromosome. I'm going to tell you about a little bit about it. So this term here is context on space. This term here measures distance on sequence. Okay, this is a very important term that we have figured out that comes into this model, and I'll tell a little bit about it. Oh, just let me tell a couple of things about it. This term here is actually a soft polymer. These things have to be able to cross a little bit. Remember, DNA is being cut all the time, right? And it's basically, we put a number that say for typical physiological conditions, like having a barrier of about 4 kT to cut, but you can put anything you want in there if you want these things to come along. The ideal chromosome term is a very interesting term. We just noticed when you do these things, you have this interaction sequence that tells you that beads that are closer in sequence, they tend to come together. And if you notice, if these interactions in sequence tend to form helical-like structures, right? You can go to protein folding, II plus 4 give you alpha helix, right? So they tend to give you helical structures. So what you see is these terms give a situation that actually, if you look on the next slide here, you just run this term, you have the sort of helices of helices, and you can see that you start to have one helix and then a helix of these helices. You start to observe that you are in the interface, where most of my presentation will be the interface. You're going to have a very well-defined helix. But if you go to the mitotic state, you start to see you have two helices. The system really becomes these big fibers where these helices form helices of helices. And you can actually, when you run the Fourier transform, to see a very well-defined, a short pitch and a long pitch helix coming to the system. Okay, so this term actually, in my point of view, there's a lot of argument on that, but we have doing these numbers. I think it's very important to avoid knot information. This sort of, if you maximize local interactions, these local interactions sort of help on not knotting, but that's not something I can tell you that has been mathematically explained away, but makes a lot of sense. So if you continue this game, so what you have on this model here, we just got the first work that we did. That time, Michele Di Piero was the postdoc in the group. You can see also his slide, right? See his initials here instead of his full name. So Michele was the postdoc on the group, and so what he did, he got this Hamiltonian, and we tried to teach this, try to learn just from chromosome 11. So we want to chromosome 11, and the reason we use chromosome 11 of GM1278 of this lymphoblastoid cell is because of the one what we saw, we had many more patterns that you could have about. Chromosome 11 is going to have about 25,000 base pairs, and on this unit it's going to be about 2,600 of these beads. So it's a good size, and we could learn these parameters from it. It's more towards the center. Okay, so it was a good chromosome, but we just look for features, right? We went around on what people learn on it, and it becomes a very interesting story. Okay, so
essentially it's sort of a local structure, basically.
On our potential, you're going to see that this interaction, if I show this potential, tells you the beads up to about 2 or 3 beads have a stronger interaction, then it goes down a little bit, gets weaker. Okay, but you can train all the way down. We have trained until maximum 20 beads and max 200 beads. Actually, when you put this cutoff, you're going to see a little bit what if I can show you something you're going to see. So anyway, these things are learned conventional way. I don't have to go into the details. There are many other different moves being done right there now. We just use our conventional technique. Now, what is this? We get the entire. every one, I think it's going to be for covariance. The first one is the size. And all the other bits are the so they are looking at the experiment by looking at how strong the signal is. The corrections, but also in the covariance of that. Things we had all the tools, exactly on a plot model, all the things we did on covariance, prediction, so but not make it between the and actually basically even sometimes the of that chip, there's lots there because for some cells you can enormous amount of chip-Seq water to get that few of them. So it's
okay, I'm just learning some of these more molecular things now. So these days, with high resolution, ChIP-Seq and other things, people use this program to decide how different.
okay. So this is not different because I just get the data from them. I just go and say what the marker. The problem is ChIP-Seq time when you now we're getting so very simple explanation there. If you really want, if you're really happy with A and B, you almost can just look at the degree of methylation. You can do a threshold of methylation. If you have a higher level of methylation, you are
Right. So how many parameters do you have here? Just a rough number, because it seems like 1, 2, 3, 4, or 5 comes there, right? And
oh, here actually, let's put it this way. This term, the best here's the matrix that's going to be 6 groups is going to be a 6 by 6 matrix, and it's just one you have to figure out how fast it falls. So there are very few parameters. But this interaction is just 6 parameters, but they're just 6 times.
this alpha KL, right? Yeah, so 6 times.
Okay. So this one here is a more interesting one because there's an entire game being played on this part. I'm going to have time to tell you that basically also you want to write chips information to that are complete. So there are lots of predictions. We have people A and D, more data, you use the transformer, then you get more correlation, then you get interesting other. And then they use the information and the types. Okay. This becomes a very interesting story because here is an interesting result, just to show some results we have at the end after you train. You start to look, for example, that this is what we predict, we call megabase. And this is high C. You see that sometimes you get things, there's a bit of mixing between the A's and the B's, but on average, you do quite well. Okay? And the great thing about being work with polymers, and I have been working with protein for a long time, is that because of the constraint of the polymer, you can tolerate mistakes. Because it's okay, right? Okay. Do that very carefully. What we do is the following: we figure out what's our level of error, compare the same level of error, and we're more or less about the same time. There is a 10% error by everybody, because a couple of things to remember, and I haven't told you guys, is that the high C. So experiments, how they cut, how they do it. So there are a lot of things we are learning these days that you have. They're different enzymes that cut to different places, but people are getting better. That's why I think it's beautiful to join in the field right now, because there's a lot of stuff being learned in real time, and part of it's by back and forth here in the Now, continuing on that, I'll tell you now, like I said, we have a tool where you can come, you start from here, and see you really have a And I'll tell you, that's something we have a lot of conversation with this, because there's a lot of people that want to get IC data that's about 1 million cells. And you get a perfect one, but that's wrong, because actually you have a big many structures, right? In fact, to do that, it's a very interesting looking data. It's a stable distribution, and as we comparing ensembles itself, what we're going to try to actually you have the basic ethnic chromosome of the group that okay, because we have chromatin, we have to go to the surface to bind the membrane, but at the chromosome level, want the gene to be expressed at the surface, otherwise they cannot express, right? And B tends to go there. But it's really a mixture. It's not like you have all A's and all B's. It's really a mixture, but there's a tendency, and this is, you can see from the colors here. And also see that these different chromosomes, here's just a simulation of two chromosomes, they tend to create compartments between them. Okay? So these things you start to observe these days. And, uh, you get start to observe. Chromosomes come together. Now, the chromosomes have a sequence, they don't have knots. What I'm telling you is that level of complex feature for all the other chromosomes to show the right feature. And then in the end, you can actually all the different chromosomes do the comparison. Here's the diagonal term. What you observe is that basically chromatin is very strong diagonal. And all the getting full sequence for chromosome of all the different species where you got the DNA. And the thing is, it's very easy to do it with a reference. You can align for the other species, you don't have a reference. So all the ChIP-Seq thing to do now, where she does chip sequencing with lots of mistakes. And then what happens is they try to do an IC map with whatever sequence she got it, and their IC map starts getting blocked on the diagonal. They start to fall in the middle. But they play Tetris with those blocks. In the diagonal, and that's a very cheap way of actually correcting all the misalignment. And that's very nice to do. But actually, if you look at that, using this idea. Here we just put very areas. And actual but she's still happy, but the thing now is basically I'd love to show that slide because it tells you how well we are doing basically. So for example, the Pearson correlation around 0.6, 0.7, as you get far from the diagonal, it's actually into different experiments. But that's enough to give the structure because there's so many ways you can do it. And see that things do really well by many other cell lines. So now we have people work with many different cell lines, different species, try to figure out when.
possible.
So it's kind of a nice group. Oh, it's kind of and actually we have gone anything and it's very good for GPU. People think they're much better how to do these things. The AMD people have it's a force prediction again. It's all and right now, 40 lines field still available, and actually we have people and work with some of our Stanford collaborators on things can get data. But the data just can't get out. It's actually getting very nice. We change. Now, one of the things we get. The problem now is everything's always like I told you that none of my potentials are trained on each. Right? But now I can say things are trained on high C, I can go to the imaging area. And actually, this the first great paper, several great papers, the first paper the group of what they on started in 2018, when she put the data, and she came a call and we were talking to each other, and she said, I'm doing imaging of the configuration compare very well with the experiment. If I can get what I have on the next slide. Okay. So here,
Sorry, before you go on. So since you have an ensemble, is there kind of like the most likely member of the ensemble?
okay, here is experimental traces structures, simulate structure. Here's the data. Okay. You can see what you want. So what you observe is that basically the experiments they have about 1,000 structures, and you have about a couple percentage of them that are the open one. We get a little bit more out of the simulation, but they are very similar. That's what we get. Okay, what temperature we use is when we try to do the system, the simulation experimental the structure. Yes.
Predict which going to be the dumbbell and which one is going to be.
Oh yes, Yes, that's no problem. Like I said, this is the data. You can actually still have both. The other one you don't have is now the nice thing about basic is that you can compare everything here and then open one here and yeah, so we're very happy. Experiment, our temperature was the actual sample we were looking at experiment, but you have to be very careful. There's no real thermodynamics. These are active processes. But what's nice about this one is if you observe this particular one, it's a very active gene, and they have all cluster on the linker. That's a very good thing. So this really appears that we're exposing genes that are exposed that genes. Why segment 2 is don't have a protein part, there's no gene on that area, and we don't observe. Let's be honest, these are observations. These are observations, but we're happy with those. And it's probably story. Big collaboration with two great in the Dutch Institute of Basic Integrated Office. So the idea now is every book is all different species. And like I said, because of the DNA2, now we got all the sequences for all the different species. So we're just now borrowing all this information. And I think this one of this paper, for example, we really like it because this is the kind of thing they say is not a theoretical paper, it's experimental paper. It's really something that just happened by having both groups work together. Like a couple of years ago, and then you have the ball now, and you have here is that group of cells, you have chromosomes. If you have water species, or axis cluster together. So that's all 5 1 etc. That's together from every chromosome. I'm going to mix the chromosomes. And a bunch of different species and we have been observing that. So when you start to work on this problem, people start to observe that also some experiments that basically that's what from brain Rollerday, that's how we got involved on that. That means you get like a human cell, you have the chromosomes well imaging, but as soon as they deplete condensate 2, that is one in time they make the system condensate, you start to mix the chromosomes. So it got very curious that actually, and remember condensate 2 is very important for this loop formation. Phase separation, all taken on back of the dilemma. So single chromosome, they could just collapse, but it's a uniform collapse, not worry about A and B different separate chromosome collapse first central don't need that. So what he observed is that basically equation. And basically, and the idea is like the chromosome is local ability can do with local groups. So we expect that term to be the one that's more strong to depend on the condensate too. Right? But basically, this ideal actually stored for nothing before. This is a term that comes from real, it's a real term from motorization. So you can actually play with that term by turning and turning on and turning off the motor, right? Basically. So at a real, when you go to 50 kilobase, you can have an effect. It is an active process at smaller scale. So putting these things together, you observe that basically you see that if you start with strong on the model, here's an ideal model with strong. You can see each chromosome here forms the very well-defined. Whereas if you start to weaken this space, you start to see now in the range of directions, you start to the central years together, and we start to now they on that thing. I remember the people were doing the press release, the rice. There was a guy there, a lot of groups press release. Yeah, right? Just by depleting. And details. There's some given analysis, and here, people are doing that. Some Daniel Association of Brazilian, and then go to all these things. And what you start to observe is when you look at the high schematic of the mosquitoes, they have these things where centromeres come together. Telomeres come together, but centromeres don't. We don't. And you get one end, you get more like much more, even so that more like yeah, something better than data 8 database. All batch version. If you start to bullet, you have to start break by the end. Start to look forward the end. But it really doesn't form the two structures of truth. Forming. Vice versa. There's lots of questions today. I think it's left, but it'd be interesting to change. He thinks that they be more mechanical sensitive, spread. Or it's going to be interesting to become mechanical sensitive, and there is an interesting question how much you just have mechanical effects at the level of the cell or just at the level of the nucleus and how the thing gets. We don't really know about it. And this is like not right. I mean, this is like but that's what it starts with. We have been doing all the prediction and hoping to get. Actually, we present one of my students. These they have very, very, very active part of genome. that will be mostly on telomers, right? Very narrow in the middle. So, and these people, like I said, we have some experimental and other teaching in French. The French are really dominating. That's why I write this proposal together. It's sort of an interesting story to tell, but it's something where you can invert on the system. We can do all the work, and there's a lot of stuff to be done. And just maybe you can either keep the pace, but it's again a system where you see like central mirror polarization in this sort of a, when you get to this sort of a, and they have the higher cell cycle. So to finish it, more tell bodies, the interesting question that happens in this field is how much you have equilibrium here and how much the system has memory and no equilibrium. And typical times appear to show that it's a single chromosome that they move. They can do it, but the entire cell nucleus cannot do it, right? There's not enough time for all this chromosome to be arranged. So the issue might allow us to actually get all the time scale. They have very precise experiments, see how much this chromosome move around. So it's a nice system to go to this part of no equilibrium part of this problem. So with that, I think I'm getting over time. I think you guys deserve some. Thank you. I think you might be.
Questions? But
Yeah, so lovely talk. I have a question about cell cycle, and what's known about how things change as you go through this, as particularly in metaphase. Because presumably the topology of the thing must change quite a lot.
Okay. So that's a very interesting story, and I'm going to look bit of what we know and what we don't know. Okay, on human cells, since when you're the interface, most of the motors tend to be cohesive, and they also have a little bit of condensate too. As you go to the metaphase, you have a major increase on condensate. It's expected because the things are getting condensed, and condensed one comes together. So you start to see a lot of higher level conversation change. These are things that remember they remember true. I could go, hey, if you want to get to human chromosome, but actually it's much easier because you can actually do it. So that's
two questions. One of which is what the relationship between what you were talking about here and what Vivek was talking about today, in which he had separation between
okay.
and so that's the first question. And sort of the second question is follows up Tani's question, which is to do with the change of structure as you go through the different phases of the cell division. And presumably you've got a changed effective interaction. Your Hamiltonian is presumably, I mean, as I gather from what you're saying, you're supposing quasi-equilibrium, some kind of adiabatic changes. At least you've got your fingers crossed and you're sort of hoping there, but somehow or other, you've tuned this potential. And I've discovered what this potential is, but how can you have discovered all of it?
Okay. You have not data in your model. And how do you know if your model is correct? Look at data. I don't know if it's correct. What you do is in the current data, you do a model, and do a model, and I do a prediction. That's why you have to have the experiments together. And you see if that model is sufficient for the effect to observe. And I even made a joke with them, say that's very funny that question comes from a physician, because when you see patients, how do you know you have enough information to do a diagnostic? And I said the difference between me and you, if I'm wrong, I don't kill anybody. But so point is well taken. So the first thing you're going to do a model like you said, in a very clear way. I'm going to solve quasi-equilibrium. I'm going to use the time scales of simulation that I have on every single stage. And then I may try to move my potential adiabatically, straining one stage. No.
Follow-up thing that is related to the different what you were talking about. So on this polling thing, I do think that if you did a more global deformation, it would be experimentally much more accessible because deforming, more global nuclear deformation is very relevant. Than polling.
And I think there's a lot of time.
So it looked like Boris, and then that'll be the last. But,
yeah, I just wanted to go back. You mentioned a few times of heterogeneity, fluctuations, and etc. So of course the comparison is on the level of pair correlation. Doing a comparison or learning for that matter. So,
and we're on the right. They're related. We compare the density of A's and B's, and we have several. Okay, the question at the end. The question is, what the function, what the geometrical feature that's important for function of these ensembles? Okay, I showed the kindergarten here on a particular segment, open and closing, but that's you're completely right. And the question is, in the end, is basically which level of comparison makes sense when you look at functional aspects.
Okay, let's thank Jose again and